I'll be nice. All right, guys, welcome back. Brand new topic today. We're going to be covering DNA, and y'all get the gist. Considering that a lot of people aren't here, if you really want your questions answered, post them in the comments or check your review again. Anyway, so let's get started. Before we continue on with DNA, we need to understand the history of DNA, how it was discovered, all sorts of shit, right? So, number one, we know a dude named T.H. Morgan, right? He is at the start of our historical journey, right? And if you don't have your notes written down, I suggest you write them down or print out a copy of the review sheet in case she sets us on lockdown browser and you cannot tab over. T.H. Morgan, this dude associated a phenotype with specific chromosomes. So he concluded that genes are on the chromosome. All right. So that is T.H. Morgan. For now, he doesn't actually know anything else other than the fact that genes and chromosomes are related. Then we get to Frederick Griffith. Now, Frederick Griffith, right? He's the guy who does the pathogenic bacteria in mice thing, right? He injects, bacteria with a strain of specific bacteria, see if they die or not, right? And here he discovers the transformation principle. And the transformation principle basically says that organisms can absorb foreign DNA and incorporate it into their own system, right? So we can incorporate foreign DNA, right? So if a bacteria comes into contact with a dead form of another bacteria, it can absorb its DNA and become pathogenic. That's what he concluded from his study. Right? Remember, he uses a neutralized version of the pathogenic bacteria. He then injects the mouse with a different version of that bacteria, which shouldn't kill it, but then it absorbs the DNA from the bad bacteria and it kills the mice. If that's unethical, don't worry, they're just mice. We don't care about them. Next is Avery, McCarty, and McLeod. McCarty. Yes, that is how you spell their names. Last names, at least. These three dudes made a very simple conclusion. DNA is genetic material. You should know these guys from, what is it? Oh, wait, no, that's the next set of guys. Anyway, these guys confirmed that DNA is genetic material, right? Then, after these guys, we have Hershey and Chase. These guys are the guys that do the radioactive experiment using bacteriophages to confirm that DNA is in fact the genetic material, right? There's another person. So these guys also conclude the same thing, but they use the test, right? They use the radiation test. Now, if you don't remember what that test is, it's basically they used an isotope or a radioactive isotope to mark the protein and then a radioactive isotope to mark the DNA. And when they discovered that the radioactive isotope that marked the DNA was the only thing that stayed in the bacteria that was infected with the bacteriophage, they were able to conclude that DNA is genetic material. So then we get to the next set of people. And we have Chargaff. And Chargaff composes Chargaff's rules. he discovers that A and T are in similar ratios, and C and G are in similar ratios, and he gives them names, right? He's the guy who discovers the existence of the nitrogen bases, but he doesn't put them together as ATCG yet, right? That happens later. Then we have Franklin. And Franklin is the lady who is able to use X-rays to get a picture. Yeah, to get a picture of DNA. So just remember, Franklin is the lady that gets the picture. And then lastly, there's Watson and Crick. They're the important guys. They discover that DNA is in this shape using Franklin's image. They also discover that A bonds to T and G bonds to C. They discover that DNA runs anti-parallel, so one will go this way, the other will go that way. And I think that's it. Yep, that's it. So y'all understand all the history stuff, right? Fairly simple. Nothing that I need to worry about. Good. So now let's get into the structure of DNA. DNA, as we know, composes of 4 sets of bases, A, T, G, C, right? Your purines are A and G, and your pyrimidines are your T and C. Remember this is important. Remember that A bonds to T with 2 H bonds and G bonds to C with 3. There's a double bond on A and T, there's a triple bond on G and C, and that has to do with structure. So, now that we remember all the basics about DNA, let's do some DNA replication, because that's going to be the first thing that you're going to be covering. DNA replication. To do DNA replication, it's pretty simple, right? You take a big strand of DNA, and you must separate it into 2 parts. Here, actually, there's a color for the second strand. Now remember, when replicating DNA, I need to remember that it is semi-conservative replication, which means when I replicate this DNA, right, I'm going to have an original strand and a new strand on both. That's some ugly looking DNA. I apologize for that. But this is semi-conservative, right? This means there is an original strand and a new strand in both sets of DNA when it's replicated, right? This was discovered in 1958 by Messelson and Stahl. You should have that on your review, so I'm not going to talk about that. Anyway, so there are 3 steps to replicating DNA. Step 1, I need to unzip DNA, right? DNA is in this shape. I don't like it in that shape. I can't replicate that shape. So what I'm going to do is I'm going to turn it into this shape. And when I turn it into that shape, right, I need what? I need an enzyme. This friendly enzyme doing the unzipping is called helicase. Now, helicase, it just unzips the DNA, right? We're going to cover what zips it back up later, but that's step 1. I need helicase to run over all that DNA strand and unwind and unzip it into this big thing so that I can actually take a look at it and, you know, replicate it. Now, we must remember, when DNA runs anti-parallel, it is because it runs in a 5' to 3' direction. What do we mean by 5' to 3'? Well, on your DNA, you have a phosphorus, that pentose sugar, because it has 5 sides, and then you have a nitrogen base, right? Now the bases bond together, so we don't need to worry about that, right? But off your sugar, there should be an extra end there that allows it to bind to another phosphorus, right? Remember, your phosphorus is what represents the 5' end, and the extra hanging off base represents the 3' end, right? So that's the direction it must always go in, because 5' must bind to 3', meaning if I have 5' here and 3' here, that means this original strand of DNA is running in this direction. And since it will be the opposite on this end because they are anti-parallel, that means this strand is going this way. Fairly simple. So that strand is going that way. That means I need to replicate in the opposite direction. So a replication strand will be 5' here, 3' here, 5' here, 3' here. And I must go 5 to 3, 5 to 3. That is the direction in which I will replicate, right? So now that we have the 5', 3', any questions on the 5', 3' actually? No? Okay, good. Fairly simple concept. So there's the 3' here. So now that helicase has unzipped the DNA, my second step is to pair new nucleotide bases to the old DNA. I'm making a new strand, right? So what I need to do is get an enzyme called primase. Let's make it magenta. Primase over here, what it's going to do is it's going to start replicating DNA by adding an RNA primer. And come on, where did I write this? Give me 1 second. So it's going to add an RNA primer, right? It'll be a specific set of RNA, a specific set of RNA bases that basically tell, or sorry, yeah, RNA primer. It's a specific set of RNA bases that tells the next enzyme to start replicating the DNA. And remember, since it's replicating, it's gonna go this way, which is convenient for the top strand. So the next thing here, let me label this. So this is primase. Primase doing its thing. The next enzyme that must appear is DNA polymerase, and we'll make it green. Good old DNA polymerase is going to jump in immediately after and start replicating the DNA, right? It's going to take, let's say the top strand is AGCTA. Sorry, let me do this in red to not confuse people watching the video. Let's say the top strand is ACGTA. The newly replicated strand is going to go in the opposite direction, right? Remember, because this is going this way, we're going to go in the opposite direction, which means the DNA opposite of A is T. That becomes A, that becomes C, that becomes G, that becomes T. Does that make sense? Yes, 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 good. So remember, this enzyme is called DNA polymerase. And DNA polymerase is just going to go in and it's going to replicate the DNA. Now, we've been talking about the top strand a lot because the top strand doesn't have a lot of issues, right? It can go from the 5' to 3' direction with no problems. But on the bottom strand, right, it's going to need a lot of extra assistance, because when it goes from the 5' to 3' direction, helicase is still going only towards the left, right? And if helicase is only going towards the left, that means that, that primase has to constantly jump back in. And I'll show this with a picture. So do the bottom strand now. We'll leave helicase there because helicase has to go towards the left. And let us label the bottom. Labeling the bottom, we have 5' here, 3' here, which means I need to replicate going this way. Do you see the problem? Helicase is moving towards the left. That means more DNA is going to unzip continuously this way. But that means I would need to add more RNA primers, right? Which means primase has to show up here, 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 here. And instead of replicating in one continuous motion, DNA polymerase has to replicate in chunks. You'll have to do that, and that, and that, and that, right? And Simonis has a video on that if you want to look at it again, or if you don't remember, or if you weren't paying attention in class. But since it needs to replicate in those chunks, those RNA primers need to be taken out. Those RNA primers, when they're taken out, there are now missing holes in the DNA. Here, 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 and here. We call those holes Ozaki fragments. Okazaki fragments. These are the special little holes in the replicated DNA that we need to fix, right? Now, here's the thing that you need to know. Okazaki fragments only happen in eukaryotes because prokaryotes have circular DNA. Their DNA is like a ring. The helicase can just unzip constantly, over and over again, right? And they can just constantly go in one direction and it won't matter, right? But in eukaryotes, there are just these holes. So how do we fix these holes? Let me move out this DNA strand. So now we have a DNA strand that looks something like this with a bunch of holes in it. Well, how do we fix it? Well, while it's still attached, what you need to add is one more enzyme. This last enzyme is called exonucleocase, or nuclease, sorry. Exonuclease removes those RNA primers, leaving the holes there, and then it tells DNA polymerase to go back in and fill in all those holes again. That's why you don't have replicated DNA with a shit ton of holes in it, right? Because exonucleocase comes in and tells the DNA polymerase, hey, we've got missing fragments. So that's how the replication on the bottom strand happens, right? The strand that's going in the opposite direction of the helicase. So now, after all that, we need one thing to end it all, right? One fresh enzyme. Oh, whoops. Hang on. Give me 1 second. I'll put it back. So now, all that being said, we've replicated the top, we've replicated the bottom, now we just need to close it out. And the final enzyme at this end, which zips it all back together, is DNA ligase. It just rezips back the, or it, what is it? Sorry, I shouldn't draw it like this. Hang on, let me get a more accurate picture. So once I've replicated my DNA, right, I'm going to have a new strand. Whoa. And an old strand, right? Because remember, it's a semi-conservative, right? I need to have an old strand paired with a new strand. And what's going to happen is an enzyme called DNA, hang on, give me 1 second, called DNA ligase will come in and we'll zip all of it back up. So then this set of two is going to come this after it's all gone through and zipped it all back up. And look at that, it's a normal looking DNA. The same thing's going to happen for the other two strands I erased, but that's what it's going to look like. Any questions on DNA replication? If you showed up late, just watch the video later when I post it. No questions, good. So now that we've done that, let's talk about genes, right? Genes need to be expressed, but the only way you can express a gene is through proteins. And well, how do you get the information to make a protein? Well, you need RNA, and in order to get RNA, you need your original DNA. So remember, gene expression goes in this order. You start at your DNA, then you go to your RNAs, then you go to your protein. The process to go from DNA to RNA is transcription. The process to go from RNA to protein is translation. Makes sense? Fairly simple stuff, right? So here we go. Transcription and translation. So let's do this in order, right? Let's go from DNA to RNA first. We are going to do transcription first. When talking about transcription, we need to go from DNA to RNA, right? So let's make a DNA strand. We'll call it so we have that set of DNA, and then we need a new set of RNA. And remember, when you're pairing up RNA, it doesn't exactly have the same bases, right? RNA doesn't have a T base. So when you transcribe it, you go like that, because the T base doesn't exist, right? It gets replaced with uracil. So when we're transcribing DNA, we need to separate our DNA back into two separate strands, right? Let me erase all this example stuff. Separating our DNA into two separate strands, we have it looking once again like that. Ah, it's ugly. Hang on. Right, so I've unzipped that DNA, and we need, and so when you unzip DNA, you remember have a 5 prime, a 3 prime end, and a 5 and a 3 prime and a 5 prime end. Remember, they're opposites, right? So when I am transcribing my DNA, I'm only actually using one of these two strands. There is a template strand and a coding strand. The template strand is being used as a template for actually transcribing it. The coding strand is the other strand. That's all you need to remember, right? How do I identify the difference? Well, remember, my RNA would ideally replicate in the same direction that DNA is unzipped, which means it needs to go this way, right? Which means your template strand has to flow in the same direction. Well, let's just mark this, because my inside, when I'm replicating it, must go in the opposite direction. It must go from the 5 prime to the 3 prime direction. The top strand is my template strand, and the bottom strand is my coding strand. What's the difference between the template strand and the coding strand? The coding strand is unaffected. The template strand is being used as a template for mRNA. That's the only difference. Nothing actually happens to the coding strand. So now that we know the parts of transcription and how to transcribe properly, let's talk about our RNA polymerases. You have rRNA, mRNA, and tRNA. mRNA being the one that we're going to care about because it's the one that we get from transcription. So when transcribing, the first thing that must happen, the first thing that must happen is a promoter needs to attach where transcription begins. On here, we are going to make this little magenta blob our promoter, right? This promoter, this area that I circled, the promoter is a section of DNA which starts the replication sequence. What does that mean? Well, remember, DNA is a series of bases, right? Oh, whoops, not you. Yeah, we'll call it that, right? So where is the promoter here? Well, the promoter is actually just right here. It's the TATA box, right? The TATA box. This box tells your RNA polymerase, the thing that needs to jump in and start replicating, to start replicating right here, right after it. All right, so the TATA box tells your RNA polymerase to start replicating right after it. It does not start at the TATA box, it starts at the base after it. So, yeah, that's essentially the promoter. The promoter is just the TATA box. Next is elongation. We've already done elongation here. Technically, elongation would be your second step. It's unzipping the DNA. Then we need to add bases in the 5' to 3' direction, which we are going to do like this. Right here in our example down here, we would notice the TATA box. Then we would start replicating right here. So then we do it becomes what is it? U A G G U. So now we have a good strand of mRNA. And then 3, our RNA sequence is going to continue until it hits a terminator sequence. What is a terminator sequence? Well, there's a ton of them, right? But essentially the terminator sequence says stop. In which case, we would have a terminator sequence right here in whatever series of characters, and then when the RNA polymerase reads that, it'll just stop. And then we have this strand, which will then flow out of the nucleus. But before we can actually send it out, it needs, well, a couple things, right? It has a 5' end and a 3' end. And remember, this end is technically the 3' end, and this end is technically the 5' end, because we went to the right when we were replicating, which means that this end is the 5' end, and this end is the 3' end. And on the 5' end, what we need to do is add a 5' cap, which is another series of bases that varies, but it protects the mRNA. And on the 3' end, it just gets a ton of A bases at the end of it, also as protection, but also as something that the ribosome is able to read to know, hey, I'm replicating this, right? Or I'm going to turn this into a protein. Ooh, sorry, my throat hurts. But yeah, so that is all that it takes to process mRNA. But now that we have a fully processed mRNA, we need to do some mRNA splicing, right? So let's take a strand. So let's take a strand of mRNA, right? I've been using green for these things. Now, when you replicate a strand of mRNA, what actually ends up happening is you get some sections in between, which you don't actually need, right? These sections are called introns. These sections must be removed. Why? Because you don't actually need them in the gene that you're trying to express. Now, why are they replicated? Honestly, I don't know, right? But the introns are there, DNA that you don't exactly need, so you got to take them out of the mRNA. What do we call the rest of the DNA that's actually necessary? And this chunk over here, we call this exons. These are the actual gene. And so what you need to do is splice a bit of or splice all the introns out. So the, hang on, checking my notes. So we would label this top thing as pre-mRNA. It's freshly replicated off of the DNA. It has the introns, it has the exons, it hasn't been spliced. It's not a mature mRNA, right? to make a mature mRNA, what I need to do is I need to bring in another enzyme. I need to bring in some SNPs, right? Yeah, let me erase all these words. So what I'm going to do in order to clean up this mRNA is I'm going to bring in an enzyme called, I'll make it a purple or magenta glob again. This one is called an SNRNP, right? These are SNPs. These are your small nuclear RNA that, or sorry, not, come on, sorry. Getting my notes mixed up. Sorry. So your SNRNPs are a series of mRNA, right? They're a series of nuclear mRNA that you actually need. I'll do that. That you need in order to splice RNA, but they can't do anything on their own. But when you put a bunch of them together, they form a spliceosome. This is what actually, it's a series of a bunch of these, right? So just like times 100. It's a bunch of SNRNPs which combine together to form a spliceosome, and that spliceosome is what hops on the pre-mRNA, goes through it, and cuts out all the introns. So your spliceosome will start here, it'll just go through it, it'll cut out all the introns. The bases that make the introns are then separated and sent back into the nuclear wasteland of floating mRNA in your nucleus. And then it forms a much smaller mRNA. This is one mature mRNA. Everything makes sense up to here? That is all the transcription process. Does that make sense? Everybody? Yes, yes, no questions, good. Cool. So now that we have a mature mRNA, a mature mRNA, we got to do some translation. So let's do some translating. So when you do translation, some things you need to understand is codons, triplets, and reading frames. A codon is just a set of 3 mRNA bases. For instance, AUG. That is a codon. It's just a set of 3. An anticodon is the complementary tRNA basis. So to figure out what the anticodon is, we'll use AUG again as an example. To figure out what the complementary bases are, it's similar to transcribing DNA, right? So in the same way I would transcribe DNA, I will take AUG and convert it into an anticodon. A binds to T, but T doesn't exist in mRNA, so I'm going to put U. U binds to A, G binds to C. That would be the anticodon of AUG. The anticodon we will deal with later, right? And then we have triplets. Triplets are sets of 3 DNA. For example, TTT, because I'm too lazy to come up with something, right? TTT, that's a triplet. It's just a set of 3 DNA bases. So after that, we need to talk about reading frames. When it comes to reading frames are essentially breaking down mRNA, DNA, or tRNA into codons and being able to read them. For instance, let's make a long strand. AUG Wait, T doesn't exist. UCC, GCC, AGG, CGC, right? Look at this. This is already cut into sets of the reading frame, right? There's a codon here, there's a codon here, there's a codon here, there's a codon here, and there's a codon here, right? I've divided what should be what you would normally write as one really long strand into 5 separate codons into the reading frame. The reading frame is literally just separating it into sets of 3 for translation. So why do I keep using AUG as an example? Because, well, that's if any of you remember, that makes the start, right? It is the start of everything. It makes the, it's the message to the ribosome to say, hey, start here, right? Because not the entirety of the mRNA is actually necessary for creating the proteins that express the gene that you need. And so, before I continue with that, next thing we need to talk about, is, well, we've talked about anticodons already, but what do we really need anticodons for? Well, anticodons, because remember, this is on the mRNA, right? When the tRNA comes in, oops. Sorry, my writing is messy today. When the tRNA comes in, something has to be bound to the tRNA in order for it to bring the right amino acid to the right place. That would be the anticodons. So these 3 anticodons will be bound to a tRNA receiver, and that tRNA, sorry, I forgot what it's called. But anyway, this anticodon is going to be connected to a separate protein which holds the amino acids necessary for making the chain. Now, that's why we care about anticodons. So when she asks for anticodons, just take your mRNA and transcribe it back into another set of mRNA and separate it into sets of 3. Because tRNA only shows up in sets of 3. It's meant to be read in the reading frame. So after that, then there's the APEs, right? Or EPA, sorry, because it goes the other way. So let's make a ribosome, right? So in your ribosome, you have 3 parts. Your A site. Let me do it in a different color for you guys. You have an A site, a P site, and an E site. The A site is where your mRNA will go in. So let's have our strand of mRNA going in like this. We'll just have it go across the entire screen. So your mRNA will enter, right? In this spot, there will be a tRNA molecule that holds your next amino acid, right? It will also enter here, and it will have an anticodon attached to it. And it will attach here, right? We'll have it holding like that. At the P site, right? At the P site, there will be another codon. I didn't make those codons wide enough, but who cares, right? So at the P site, this is basically just a holding spot for this tRNA that's holding the amino acid. When the next tRNA comes in at the A site, it will form a bond between these 2, connecting the 2 amino acids together and beginning to form a chain. At the E site, hang on, let me draw another protein here. At the E site, what's going to happen is, since this bond is already established, we don't need that tRNA. We can send it off to go grab another amino acid later on in the line. So at tRNA is thusly ejected. This is all occurring in the ribosome, by the way, if you were confused about that. So at the A site, the tRNA carrying the next amino acid will enter. It will recognize based on the anticodons to the normal codon if it's correct or not. Then that will form a bond with whatever amino acid is in the P site. If there is no amino acid, it'll just get pushed over into the P site. Then from the P site, once it's firmly established, it will then send it to the E site where the tRNA is ejected, but the bond holds the amino acid placed there by the tRNA just set, right? As you continue along, this chain will continue. It won't stay in the ribosome. It'll just keep going. Like so. And that's how you make an amino acid chain. All right. All right, cool. Now that we have covered the APE site, or the EPA site, let's talk about mutations. So mutations come in two different varieties, right? Let's draw this out. Oh, come on. Mutations. Mutations come in two varieties. One is a point mutation, and the other is a frame shift. Now, a point mutation only happens at one base. A frame shift mutation shifts multiple bases. Or the reading frame. That's the key thing you need to remember, right? A point mutation only affects one base. A frame shift affects multiple bases because it shifts the whole reading frame, right? So let's do point mutations first. And a point mutation. There are three different varieties of point mutation, right? But let's go in depth into what a point mutation is. So say, for instance, your DNA strand is AAA. A point mutation will cause this AAA strand to become AAC, or ACA, or CAA, right? Only one of the bases is changed. The first scenario, we have the third base changing, and the second scenario, we have the second base changing, and in the third scenario, we have the first base changing, right? Now, for the example I'm going to try to use, all three of these might not work because I forgot to pull out a codon chart, but the situations will make sense when you actually see them, right? First is the silent mutation, right? Your silent mutation is just when there's a mutation, but the amino acid stays the same. So you don't see the change, right? You cannot see the change in your genes. You can't see the mutation, but the mutation is there. This typically happens in the third base. But sometimes it can happen in the second. You usually won't see this in the first. The second type of mutation is a missense mutation, right? Where the amino acid changes. This typically happens in the first or second base, but typically happens in the second. Your third type of mutation is a nonsense mutation. A nonsense mutation just says stop. It makes the chain stop where it shouldn't stop, right? And in a nonsense mutation, this typically happens in the first base. So just remember these differences. Silent, the amino acid stays the same, but the mutation is there. Missense, the amino acid changes, but the chain doesn't stop. Nonsense, the chain stops. Fairly simple. Now let's go over frame shift mutations. In a frame shift mutation, two things can happen. One, there's an insert, or two, there is a deletion. For instance, if I have an M or a DNA strand that says A, T, T, C, C, G, G, right? And I insert a new mutation where I put an extra C here, right? So now my new strand says A, T, T, C, 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 G, G, right? I have inserted something and the frames have shifted, right? Because in the old one, the codons were A, T, T, C, 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 G, G. But in this new one, it is A, T, T, C, C, or wait a minute, A, T, T, C, C, G, G. Oh, wait a minute, have I screwed this up? Yeah, A, T, T, C, C. Oh, wait, here we go. That's where I messed up. There shouldn't be a C here. It should just be GG, right? But now I've shifted it, right? So now that now instead of having 2 codons, I now have 3, meaning something here is going to be produced that shouldn't have been produced. In a deletion, let's use the same strand, A, T, T, C, C, G, G. And let's say, for instance, I delete this C. Now, what I get is A, T, T, C, G, G. And my codons are A, T, T, C, G, G. And this is, or sorry, where it should have been, sorry, my mistake. It should have been T, C, C, and G, G. This is now A, T, T, C, G, G. Notice, sure, I have 2 codons, but the middle codon is completely different. So now the second amino acid that I would normally put here is completely different. That's the difference between a point mutation, a frame shift mutation, and all their special things, right? Frame shift mutation only has the 2 situations, the insert and the deletion. Just know that it changes the reading frame where the codons are, which the codons are in. Last thing we need to cover is viruses. Viruses, just a couple key points on viruses. They are non-living, gotta remember that. Good old viruses not actually being alive, but they have DNA or RNA, some sort of ribonucleic acid, right? Some sort of genetic material. They can adapt. And most importantly, they can replicate. Now, viruses come in 2 different shapes. You have your macrophage or bacteriophage, which has the weird spider legs on it. And then you have your coronavirus, right? Where it has the weird receptor things on the end. Give me 1 second while I finish this shape. Oh. Right. When labeling these shapes, just remember, or sorry, the genetic information is there and there. Remember, the bacteriophage has a protein capsid, which is its neat little head. And these receptors on these more circular brain-shaped viruses are called glycoproteins. And those glycoproteins allow it to invade a host cell and start replicating. And so, next point about viruses, viruses. Viruses come and go in 2 separate cycles. One is your lysogenic cycle. In this cycle, it the virus will send its DNA into a host cell and it will just sit there and wait for the perfect condition. So this tends to be dormant. You tend to see, you tend to have this over a long period of time. Your symptoms don't show until later, all sorts of stuff, right? But once the lysogenic type virus enters its kill stage or replication stage, it will do the lytic cycle. And the lytic cycle, remember, these are cycles. The lytic cycle is essentially the short form where symptoms appear very quickly. And the virus begins replicating itself. And the virus will essentially invade the host, give it part of the viral DNA, trick the host into producing the virus constantly until the host goes from being normal to being completely full of big virus man. Will make it magenta globules until our poor cell explodes. And out comes the virus. Pretty simple stuff. The lytic cycle, remember, lysogenic, longer word, longer cycle. It's dormant. It takes a long time. Lytic cycle, shorter word, short. Just make the cell go boom. Other than that, that's everything. Cool. If anybody's actually listening to me, any questions?
No.
Ah, good. People are here. That's neat. All right. I will get this video posted very quickly. Outside of that, since I have to do a stereotypical YouTube outro, everybody say hi, YouTube. If y'all want to.
Hi, YouTube. YouTube.
Matthew, you want to say hi? All right. I hope that caught him. Anyway, thanks for watching. If you have a question, post it in the comments. If I'm up at midnight, I will try to answer it. Godspeed to all of you. I know most of you are going to be cramming late into the night, but remember, get some actual sleep. Especially if you have bio first or second period, otherwise you're going to die. Other than that, thanks for watching.
Thanks, Marcus.